Isaac and Colin, and we're here for Waterloo iGEM today to talk about our project for this year, Prions Be Lit, Functional Amyloid as a Biological Tool. So just to give you a little bit of an overview, first we're going to go over the background of our project and some of the parts that we're using, and then we're going to talk about the math modeling and some of the theoretical considerations that's going to flow into the policy side of our project, and finally we're going to go over some of the results that we saw in the lab. So first off, I guess, what are prions? So prions are infectious particles made of aggregated proteins. Typically this is the same protein which is aggregated with itself. So they're found in nature in a variety of places. In mammals, they're often found and associated with diseases, but in some animals, as well as plants and fungi, they're actually just a normal functional part of the cell. So these are called functional prions or functional amyloid, which is just a broader name for a kind of protein aggregate. So an example of a functional prion would be sub-35, that's found in yeast. And when yeast has sub-35 just floating around in a non-aggregated form, that cell is called Psi-. When it's aggregated together, the cell is called Psi+. So the structure of sub-35 is that it's got three different domains. It's got a prion domain at its N-terminal. That's responsible for its prion activity. It can take one of two forms, one of which makes it aggregate. It's got a charged or a middle domain in the center, and researchers aren't entirely certain the purpose of that. And then it's got a functional domain at its C-terminal, which is just responsible for the normal function of sub-35 in a yeast cell. So the cool thing about prion domains is that they're modular. What this means is you can take sub-35's prion domain, and you can attach it to a different protein, and give that protein the ability to aggregate like sub-35, and it will actually aggregate with sub-35 if you express them together. So our goal is to use that as a kind of engineering tool to bring different proteins close together in or outside of a cell. So basically what we're trying to do with that is to encourage protein-protein interactions. And the way they've defined them, there are two different kinds of protein-protein interactions that you can get. So you can get them an interaction through them just being close together and, say, passing off a substrate from one enzyme to another. Or you can actually get them physically interacting. So we've designed two different experiments to test those two things. We're going to use force or resonance energy transfer to test proximity, and bimolecular fluorescence complementation to test the ability to physically interact.
So our first proof of concept is FRED, or force to resonance energy transfer. So basically how this works is you have two fluorescent proteins, in our case cyan fluorescent protein, or CFP, and yellow fluorescent protein, or YFP. And CFP actually excites YFP. So when you excite CFP at some wavelength of light, you'll only see a yellow signal when CFP is sufficiently close to YFP so that it can excite it. So the hope in our experiment is that in the Psi+ state, or when they're starting to join the aggregates, that'll pull them close enough together so that we'll see this increase in a yellow signal. So the way we went about this was we used a combinatorial model. So combinatorics is a branch of mathematics concerned with finite structure of things, or enumerating how different things can form, specifically discrete structure. So in our case, this made a lot of sense because we're looking at effectively strings of proteins of a finite length. So what we did here was we looked at, based on the length of an aggregate, the relative efficiency, which is sort of a measure of the average number of YFPs which are being excited as a percentage of the length of the amyloid. So as you can see here, it sort of looks like it's converging to some value, maybe about 0.225, and we don't know what this value is. Part of the reason we don't know is because the equation that we derived for calculating this is horrible. This isn't even a closed form equation, this is just for computing sums, so we plugged this into another Python script to actually get that previous slide. So we could only get up to 15 length amyloid before the computation just became too intense. So our other proof of concept is called BIFC, bimolecular fluorescence complementation. And that's where what you do is you take a YFP, so a yellow fluorescent protein, and you cleave it in half. And then we attach each of the two halves with a prion domain. So when they get sufficiently close together, they can actually snap back and form a complete YFP. And then again, you see this yellow signal. So again, the hope is that in the aggregated state, in the Psi+ state, you're pulling them again sufficiently close together so that they can snap, and then you'll see an increase in yellow. So the first thing we did here is we created an ordinary differential equation model. And sort of the purpose of this was we wanted to be able to tell the ratio of free-floating YFP halves, free-floating complete YFP, and YFP which is involved in aggregate. So our system only affects the YFP that actually joins the aggregate, so it's critical that we can understand this. Then we proceeded as before with a combinatorial model. So the rules were a little bit different, but basically what we're doing is we're measuring the amount of YFP, which is in a conformation such that it can form pairs. So for instance, if you have all of one half on the one side and all of the other half on the other side, that's very bad. Ideally, you want them flip-flopping. So again, here, it sort of looks like it's converging to some value, maybe a third. And in this case, we actually know it's a third. So we know this for two reasons. One, we created a Python script to run the simulation hundreds of thousands of times and come up with an average. And two, we use this technique called Markov chains, which is a probabilistic model that tells us as the aggregate grows infinitely large, what is the steady state ratio of proteins involved in each of the different states. So basically some takeaways from this are, it's optimized when there's no sub-35. So sub-35 doesn't interact with any of our fluorescent proteins, so it only interferes with their interactions. It's optimized when the concentrations of two YFP halves are equal. The efficiency of the system gets small as the relative concentration of any of the three proteins grows arbitrarily large, so that's sub-35 or the two halves of the split YFP. And then finally, the efficiency hit from introducing sub-35 is, it makes it worse, but it doesn't make it so much worse so that it's worth engineering what we're calling an orthogonal prion domain, which would be a prion domain that doesn't interact with the native sub-35 in the cell. So one of the important applications of our system is this thing called substrate channeling. So that's where you have two enzymes and they pass a substrate to each other. And so the efficiency of that is related to the time it takes from the substrate to transition from the first enzyme to the second. So this is very similar to what we're doing with our FRET experiment, where they're passing a photon from the first enzyme to the second. So what we were doing here is sort of creating a transformation between data that we could measure from our FRET experiments to be able to make predictions about the efficiency of a theoretical substrate channeling system.
All right, so I'm going to be talking about what we did with the human practices team. And in human practices, we found that the results that we gathered was integral and central not only to what the wet lab was being designed, but also the data that we collected and how we interpreted it and applied it to not only all of our stakeholders, but also to the public and to our experts that we interviewed as well. So as Isaac mentioned previously, we needed to have a lot of applications in order to help understand our foundational advanced project and communicate our foundational advanced project to the public. So these are 6 different auxiliary applications that we were able to come up with as well, based on the interactions we had with experts. These include alternative fermentation pathways to produce ethanol based on different metabolic pathways, antimicrobial peptides that you can create to combat against superviruses and superpathogens. We can apply our protein aggregates to apply them to nanosensors. We can have alternative ways to have peptide purifications. We can have targeted apoptosis, first of all with prokaryotes, and then make our way up into different systems, hopefully to human models as well. And then also vaccine preparations as well, in terms of reducing the number of doses and administrations that you can have in your vaccine. So one really cool thing that we were able to create is something called the Implicit Association Test, or IAT. And what we were trying to accomplish with this was having a psychological tool to determine implicit biases that people have towards synthetic biology and towards our project in general. The way this worked was that we had different words come up onto a screen that were either related to synthetic biology / biotechnology or traditional technology. And each person would associate this into a category of pleasant or unpleasant. For instance, the word prion, which is key integral to our project, would come up onto the screen. People would associate it under the key of perhaps biotechnology and then pleasant, which is actually what we end up finding as well. Another dynamic way that this test helped understand people's biases was the measured time in order to respond to it. If they respond to it fast, we understand that they have a certain bias towards whether they think it's pleasant or unpleasant more. So really cool results that we found was that the word prion was associated with very fast response time, and it was under the pleasant section. This is very encouraging to us because it helps people know that they understand our project and they think that it's safe and ethical as well. Words such as yeast and aggregate were actually found to have very longer response times and had no clear split towards whether they thought it was under biotechnology or traditional technology, meaning that we may have further tasks to do in order to help communicate our project further to the public. Some other ways that we like to take this to the next step would be to have a greater sample size and also to really have a more diverse background of people taking this test. Another really cool application that we were able to create with our project is a game called Snake 35, which is a take on a classic game called Snake that I'm sure many of you have played. This was a perfect way to help us analyze and gamify our Fred experiment, which Isaac alluded to previously. All you have to do is collect your YFPs and have them right next to your CFPs in order to excite the YFP or collect points. We use this game at many of our outreach events, to which I will describe later, and have received a very positive response from it. I encourage you to go onto our wiki and play it and try to beat my high score of 66. In public engagement, we did 3 very large events that we ran that were very diverse between each other. We had a Shad Valley workshop in which we took high school students from across the country and talked to them about ethical principles within synthetic biology, and also ran through very basic synthetic biology experiments. We had a gender equity panel where we took experts from many different fields and talked about the difference between gendered minorities and how we can reconcile that within the field of synthetic biology. And we also held a science open house to which people from many different backgrounds were able to design their own microbe, and we could teach them how we designed our own microbes as well. We have a lot of other auxiliary projects that we'd like to tackle that I'd like to touch on very quickly for you guys. So for our silver medal criteria, we had a project determination chart in which we used in order to determine which project that we would have, which project that we should choose back in February. We were initially deciding whether or not to work with Prions or not. We had a policy guidebook that we created in order to understand how to create a policy team within iGEM, and then what projects that you can create within your own policy team as well. We had a lot of 3D printing that we did for basic lab equipment, such as a 3D printed comb right here. Some of them sell for very large costs, and we wanted to bridge that financial cost by 3D printing them for as low as 50 cents. We created a documentary called The Gem in the Rough to discuss a lot of the different teams within iGEM and how we formulated our project. And we went to something called a biotech boot camp as well, where we really tried to understand how to ramp up our social media game and really communicate our science in the most ethical way possible to our teams. And we did this by vamping up our Instagram and Facebook as well. We had also 2 other auxiliary projects that conferences that we went to, including the Canadian Society of Microbiology Conference and the SynBio Symposium at Western University.
So we also had a small software project this year, which we're calling CIGEM, which is for a continuous integration server. So continuous integration is kind of a practice in modern software development where the repository that actually holds your code directly represents what is on the server. So in our case, we made a continuous integration server for the iGEM wiki where you can push your code and work on it collaboratively on GitHub, and then when it gets merged to master, it actually deploys it, so it uploads to the wiki automatically. So this means that you never have people overwriting each other's files, you never have to copy and paste into text boxes, you never have to put JavaScript in templates and things like that. So it just really streamlines the process of actually building the wiki. So we made this on top of a lot of tools that University of Toronto iGEM team had previously made, and they were a huge help in assisting us to create this year.
All right, now that we've gone over the background of the project and what Math and Policy did, we're going to move on to the lab. So first off, how we design our parts, as Isaac previously mentioned, for our Fred experiments, we were using cyan fluorescent protein and yellow fluorescent protein. The reason why we chose these 2 is because you've got a pretty good interaction between them. It's relatively easy to measure compared to other fluorescent protein pairs. So what we did was we took the sub-35 prion domain, which we defined as the first 137 amino acids, which is the sequence that's been shown to be necessary for the full prion phenotype of sub-35, and we attached that with a glycine linker to CFP and YFP. And just as a reminder what's happening here, the prion domain is going to end up in the core of our little aggregate, and then our cyan fluorescent protein is going to be kind of branching off, which is why it can still maintain its function. So for our Fred experiments, we did a pretty similar thing, where we took, again, the prion domain and linked it with a glycine linker to split halves of YFP. On top of this, we also biobricked the prion domain of sub-35 on its own in case someone wanted to create an engineered prion in the way that we did. And we also further characterized the part that our team made last year, which was the cup-1 promoter followed by GFP. So the cup-1 promoter is just an inducible promoter in yeast, which you can trigger by adding copper to your growth medium, which we characterize in a number of ways if you see our wiki. So the first experiment that we did was we took our PRD CFP and our PRD YFP, so our parts for Fred, and we transformed them into yeast cells, both together and individually, into cells that both did and did not have sub-35 aggregates. So here is a emission spectrum for excitation at 488 nanometers, so that should excite YFP. And we do indeed see that in our psi- samples with YFP, we have a peak exactly where we expect it for yellow fluorescent protein. When we tried this again at 430 nanometers to excite CFP, our data ended up being fairly noisy, so we couldn't confirm or prove against that the CFP was actually fluorescing, but YFP was most definitely fluorescing in the psi- state. The curious thing here is that our YFP was not fluorescing in the psi+ state, so the state that had the aggregates. So to look at this a little bit more, we put our cell samples into a flow cytometer, which has the ability to look at cells individually, excite them at 488 nanometers, and observe if they're fluorescent or not. So through our cup-1 characterization, we came up with a threshold for what is and is not a fluorescent cell. We sorted the cells accordingly. So the numbers you see right here are the percent of our cells that we put through the flow cytometer that were fluorescent. So as you can see, our psi- cells that contained YFP were all fluorescent, but of our psi+ cells that contained YFP, one was not fluorescent and the other had very weak fluorescence. So this is actually quite interesting because it tells us something about probably the linker that we used to, the prion domain that we used to give the aggregation behavior, because what some labs have done is they've attached the prion domain of sub-35 and the middle domain of sub-35 to a fluorescent protein and observed that join an aggregate and then fluoresce. But we hadn't found anyone who had just attached the prion domain, and so with the spirit of having a smaller and simpler part, we started off by only attaching that. But it seems like that was not sufficient, so when our YFP joined the aggregate, it's lost its fluorescence. So it seems like the middle domain, there's a theory that serves as a spacer between the core of your aggregate, which has the prion domain, and whatever's dangling off of it. And it seems like our result here supports that, because what seems to be happening is our YFP, when it joins the aggregate, is too close to the center, and it either gets squished, or it gets buried in the aggregate, or it's just never able to fold in the first place, and so it loses most of its function. Which is interesting, because it tells us something about possibly the fundamental structure of sub-35 or prions in general. So this leaves us interesting experiments to do, such as adding the middle or the charged domain and seeing if we observe increased fluorescence to support this result. We can also complete our split YFP experiments. We created those parts and biobricked them. But when we did our transformation, the cells didn't grow. We can also continue this as a design project. So at Waterloo, all engineers do a design project in their 4th year. The project my group and I are going to do is going to focus on this, where we go to the next stage and try to attach actual enzymes to the prion domain and try to increase the speed of a pathway. So just really quickly, going over the metal criteria. Well, the Prions were here. We had fun. We were presenting at the Jamboree. And we filled all the deliverables. We completed our attributions page, and we further characterized the cup-1 promoter. For silver, we showed that our part PRD YFP does in fact fluoresce on its own, and it does interact with the aggregate because of this loss of fluorescence. And we had a number of collaborations with Dalhousie, Toronto, and Loaf.
In terms of our human practices for our silver medal criteria, as mentioned previously, we had a project design criteria where that had different criteria where you can classify novelty or feasibility to determine which project that we should choose. This is based on qualitative and quantitative material. For a gold medal, as mentioned previously, we had integrated human practices of our IAT test and also our Snake 35 game in order to help further describe our project as a foundational advance.
For our modeling gold medal requirement, we created 4 unique models, all based on completely different branches of mathematics. This allowed us to decide not to engineer an orthogonal domain, which saved a lot of time and sources of error. As well, the modeling allowed us to estimate the efficiency of the substrate channeling application based on experimental data that we could receive from our FRET experiments.
In terms of the lab, though we didn't observe the FRET result or the BIFC, we do have evidence that we were able to express 2 engineered prions within the same cell. So we know YFP was there, and because we know our promoter was working and our prion domain was working, CFP was probably there, and that's something that we don't think someone has done before. Here's who we'd like to thank, and our sponsors. So that's it for our presentation. Thank you very much for listening.